According to the World Health Organization, cancer is defined as a generic term for a large group of diseases that can affect any part of the body. It is caused by the transformation of a normal cell to a cancerous cell, which can be triggered by many different factors. An instance of cancer may be a result of a combination of various factors, including genetic factors or physical, chemical, or biological carcinogens. While specific causes of cancer vary, the World Health Organization also states that around 1/3 of deaths from cancer are due to tobacco use, high body mass index, alcohol consumption, low fruit and vegetable intake, and lack of physical activity. So how can fruit and vegetable intake impact our risk of developing cancer? This video will explore the role of exogenous antioxidants in pancreatic cancer prevention. First, let's backtrack a little and talk about free radicals. Free radicals are molecules containing oxygen that are unstable due to an odd number of electrons. They are produced as a product of normal body processes, but may also be derived from external sources, like air pollution, cigarette smoke, UV rays, and X-rays. So here we have an O2 molecule, for example. If we split this molecule, each atom becomes a free radical, hence why oxygen is a diatomic element. In order to become stable, these free radicals steal electrons from stable molecules in the body, which then become free radicals, creating this cycle of free radical formation. When functioning properly, free radicals can attack harmful substances in the body. However, when the cycle of free radical formation is left unmediated, the disturbances caused by free radicals may create a condition of the body called oxidative stress. Oxidative stress can cause damage to lipids, proteins, and DNA, which can lead to health consequences such as autoimmune disorders, advanced aging, cardiovascular and neurodegenerative disease, and cancer. To maintain a balance of free radicals in the body and prevent oxidative stress, we need antioxidants. These are molecules that can donate electrons to free radicals to stabilize them. Antioxidants, however, unlike other molecules, are able to do this without sacrificing their own neutrality. This allows them to keep free radical formation under check. There are two different types of antioxidants, exogenous and endogenous. Exogenous antioxidants are those consumed as part of the diet, while endogenous antioxidants are those that are produced by the body. Some notable exogenous antioxidants are vitamins A, C, and K, and other minerals or phytochemicals. These exogenous antioxidants are most abundant in fruits and vegetables, but can also be found in many other food products such as some meat and fish. Let's take a look at how much exogenous antioxidants have an effect on pancreatic cancer risk. For this first study, we will consider this question on a cellular level. These researchers carried out a molecular hybridization, which is the combination of two or more non-identical pharmacophores into a single molecule. In this study, they synthesized hybrids containing a well-known anti-cancer chemotherapeutic agent called 5-fluorouracil with coumarin derivatives. Coumarins are an antioxidant found in plants such as cinnamon, licorice, and apricots. These compounds were tested as antioxidants in vitro with pancreatic cancer cells against a reference standard of ascorbic acid. Their antioxidant activities were evaluated by their scavenging effect on DPPH radicals, which was measured by the bleaching of the purple-colored methanol solution of DPPH. Each graph shown represents a different pancreatic cancer cell and its response to 2 fluorouracil agents and 2 of the fluorouracil coumarin hybrids. In PANC1 cells, the synthesized hybrid CP2 had a large effect on lowering the cell viability. We can conclude that 5-fluorouracil, when hybridized with coumarins, exhibit stronger antioxidant activity in PANC1 cells. This demonstrates antioxidants' defensive roles as cytotoxic agents against pancreatic cancer. Another study examined plasma levels of antioxidants and their relation to cancer risk. The design of the study was that 446 people with pancreatic cancer were paired with controls of similar age, sex, hormone use, etc. Plasma levels of carotenoids, tocopherols, retinol, and vitamin C were analyzed. Here is a graph of their findings regarding beta-carotene. The correlation efficient for these findings was a negative value, meaning that pancreatic cancer risk and plasma beta-carotene levels are inversely related. Consequently, it was found that higher levels of beta-carotene are associated with a lower risk of pancreatic cancer. Beta-carotene was also more effective in lowering risk in non-smokers compared to former and current smokers. Like beta-carotene, inverse relationships with zeaxanthin and A-tocopherol plasma levels and pancreatic cancer risk were also found to be statistically significant, as seen here. These are all examples of exogenous antioxidants. Beta-carotene is a carotenoid related to vitamin A that is found in yellow and orange fruits and vegetables such as carrots, squash, and cantaloupe. Zeaxanthin is also a carotenoid related to vitamin A found in green plants such as spinach, pistachios, and peas. A-tocopherol is one of 8 isoforms of vitamin E found in soybean, sunflower, and almond oil. This final study is one that was structured as a questionnaire. The questionnaire was mailed to a case group who had pancreatic cancer and to a control group who had a similar age and gender distribution as the case group. A majority of the questionnaire concerned food consumption, which was used to estimate daily intake of total carotenoids. Participants were also asked about smoking history, height, weight, physical activity, and education. After adjustment for age, province, education, smoking, BMI, folate, and total energy intake, the results show that there was a significant inverse association between pancreatic cancer risk and lycopene in men. Lycopene is a carotenoid found in red fruits and vegetables like tomatoes, papayas, and watermelon. In this study, a majority of lycopene intake was from tomatoes, tomato juices, and tomato sauces. Beta-carotene was also again found to be significantly related to reduced pancreatic cancer risk in people who never smoked. Due to the function of antioxidants in the body and their role in oxidative stress prevention, it can be assumed that they play some sort of role in cancer prevention. Each of these studies covered in this video have found significant correlations between either decreased pancreatic cancer cell viability in the presence of exogenous antioxidants or decreased pancreatic cancer risk in people who have higher consumptions of certain exogenous antioxidants. However, these studies also explain that eating fruits and vegetables alone will not protect against pancreatic cancer. The discussion section of the questionnaire study did mention that exposure to tobacco smoke, which produces free radicals, causes beta-carotene oxidation and also lowers circulating levels of beta-carotene and other antioxidants. Because of this, their findings that certain antioxidants were effective in reducing pancreatic cancer risk could only be applied to people who have not smoked. The function of antioxidants within the body is complex, and their efficiency can be hindered by certain exposures and lifestyle choices. But these findings still support the fact that, in conjunction with a healthy lifestyle, antioxidants consumed in the diet may reduce risk of pancreatic cancer. Thank you for watching, and eat your veggies.